Hello everyone, my name is Safia and I am here to assist you with the subject biology. Let us start the topic. But before I start the topic, I'll ask you some questions. We do a lot of activities from morning to evening. Let us say like we play, we read, write, run, jump. For all these activities we need energy, right? From where are we getting this energy from? Exactly, we eat food. We eat food to get energy. And this process of acquiring food is called as nutrition. Okay? Nutrition is the process of acquiring food. Process of food. And do we alone need energy for doing activities, or all the organisms living on this planet Earth require energy? Exactly, every organism, whether it is a unicellular organism or a largest of multicellular organisms like blue whale, every organism requires energy. So, based on how the organisms acquire their energy, there are two types of organisms. Based on the nutrition, there are two types of organisms. What are those two types? First one, autotrophs, or you can say as autotrophic organisms also. Second one is heterotrophs. What is the difference? In nature, some organisms have capacity to make their own food. They do not depend on any other organism for their food. They depend on non-living organisms like water, carbon dioxide, sunlight, but not on any other living organism. Such organisms which can make their own food, are called as autotrophs. But organisms like us, we cannot make our own food without depending on plants or animals. We need, suppose if you are making rice in the morning, you need rice grains. If you are making curry, you need the vegetables. If you are eating snacks or something, fruits or vegetables, something is required. Meat is required, eggs are required, milk is required. These are not from non-living sources, they are from living sources only. So, there are certain organisms which cannot make their own food, but depend on other organisms for their food. Okay, since they cannot make their own food, they need energy, they cannot live without energy. So, they depend on other organisms for their food, Okay. So we'll discuss these two in detail, starting with autotrophs. Autotrophs are those organisms which can make their own food. Most familiar example of autotrophs are green plants. All green plants can make their own food. All green plants can make their own food by using sunlight, carbon dioxide, water in the presence of chlorophyll. Okay. If chlorophyll is not present in a plant, they will not make their own food. Only if chlorophyll is present in the plant, they can make their own food. In the presence of sunlight, carbon dioxide and water. Okay. As a result, what is produced if these things are utilized? What is the result that is obtained? Glucose molecules are produced by plants. The glucose molecules produced by the plants are used to build a complex molecule called as starch. Okay, from starch different molecules are produced like oils or proteins. These different things are produced gradually from this molecule. So glucose, along with glucose, some amount of oxygen molecules are released, some amount of water is also released. Okay, so these are the factors that are required for photosynthesis. Okay, these are the products that are obtained from photosynthesis. These are products. Okay, so if you write them as a formula, it comes like this: 6 CO2 plus 12 H2O gives rise to C6H12. You can write it. I'll write it here. 6 CO2 plus 12 H2O gives rise to C6H12O6 plus 6 oxygen molecules plus 6 water molecules. Okay. In the presence of sunlight and chlorophyll. So what are the molecules that are required? Carbon dioxide, sunlight, chlorophyll and water. These four things are required. What are the products obtained? Glucose, oxygen, water. So this can be proved. Okay, this can be proved that by some of the experiments. We'll see how. There is an experiment to prove that starch is produced as a result of photosynthesis and oxygen is produced as a result of photosynthesis and chlorophyll and sunlight are required for photosynthesis as well as carbon dioxide is required for photosynthesis. Okay, chlorophyll experiment is not given in your textbook, water experiment is not given in your textbook. Let us leave those. In your textbook we have only four of these important experiments: carbon dioxide, sunlight, glucose and oxygen. These 4 experiments will go through. Okay. Okay, the first experiment is experiment to test the presence of starch in leaf. Okay, from where the starch comes, it is making during process of photosynthesis. It is making. So that is why it is present in the leaves. And we know very famous starch test. What is the famous starch test? If you take any substance which is having starch and put few drops of iodine over that, it will turn in a short time into bluish black color. See, this is a leaf which is having starch. If you put few drops of iodine solution over this, after a few minutes you can see that there is a change in color. It turns into bluish black color. Okay, this is a simple experiment to show the presence of starch. But we are having a problem with leaf, it is green in color. So there may be a confusion when you test whether blue color is obtained or not. In that green color you will not understand. So first of all, we need to make this leaf white color like this. How? Removal of chlorophyll. Chlorophyll should be removed. This can be done with the help of another liquid. What is that liquid? methylated spirit. Methylated spirit. If you burn it, take the leaf inside methylated spirit and boil it in a beaker containing water. After some time, leaf turns into white color like this. Okay. So let us see the steps of this experiment, starting from the materials required. So these are the materials that are required to perform this experiment. You need a leaf, a tripod stand on which this beaker will be set, beaker and also test tube. To keep the beaker and test tube, you need an iron gauge to place over this. Okay, so that is also required. To hold the test tube, you need a test tube holder, and to burn this beaker and its content, you need a Bunsen burner. Okay, a dropper to drop the iodine solution over the leaf, and iodine solution itself, and methylated spirit. A petri dish also is required. Okay, so these are the requirements for performing the experiment. Next, let us see the procedure for the experiment. Initially, you need to collect the leaf on which you need to perform the experiment. In a beaker, you need to take some water. Water into a beaker. Now, take the test tube. Pour some amount of methylated spirit into that test tube. After that, insert the leaf, this leaf, which you have to test for the presence of starch, into this methylated spirit. After that, you need to take this test tube into this beaker, and see, we have placed the Bunsen burner here. So, this Bunsen burner helps to give flame to this beaker. So it starts boiling. So it takes approximately around 1 hour to decolorize this leaf, to destroy the chlorophyll completely. Okay. Now the leaf turns white in color. Place it in the petri dish. After placing in the petri dish, see this methylated spirit turned into green color. That means all the chlorophyll entered into this, dissolved into that methylated spirit, and leaf turned white in color. Now you can go with the starch test. The simple starch test, which you know very well, you have also done this in your class 7. Take the leaf, put few drops of iodine solution over that. Observe the color, it's not bluish black now, it is brownish orange color. After a few minutes, if you observe the same leaf, you can see there is a change in color. The iodine solution will turn into bluish black color. So this proves that starch is present in the leaf. So sir, our first experiment is done. We have proved that starch is present inside the leaves. So this is the first experiment. Coming to the second experiment. Oxygen is liberated during photosynthesis. For this experiment, we need a plant in such a way that we can collect all the oxygen coming from that. If you take any land plant, terrestrial plant, you cannot collect the oxygen coming from that. So we go with the aquatic plant, hydrilla. This plant, if you take this plant into a beaker, you can clearly see the bubbles that are coming. You observe aquatic plants several times, you see bubbles coming out of them very easily. So that gas can be easily collected compared to any terrestrial plant. So we will go with an aquatic plant like hydrilla for performing this oxygen experiment in plants. Okay, oxygen is liberated during photosynthesis. That's our experiment. So what we need for this experiment? What are the materials required for this experiment are so these are the things that are required: a beaker, a glass funnel, a test tube, hydrilla plant, and a burning matchstick or splinter, whatever is. So this burning matchstick, what is the need? When oxygen is collected and it is tested, you know the test for oxygen is very simple test. Oxygen helps combustion, burning. So if something burns very well in presence of that gas, then you can say that that is an oxygen gas. So here also we are doing same thing. We'll collect all the oxygen into this test tube and we'll bring this splinter, burning splinter to this test tube and check whether it is burning well or not. So these are the materials required. Our setup will be like this. We need to take this hydrilla plant. We need to put this hydrilla plant into this funnel. Okay, insert it into this funnel like this and put it in the beaker containing water in inverted way. Why inverted? See, this all the gas coming from this will be concentrated in this region. It will not go this side and that side. So that is the advantage. Now it should not escape. We should collect it. So for that reason, we will keep a water-filled test tube into this beaker. See, when you are inserting carefully, you should be careful that no single gas bubbles enters. So close it with the thumb carefully. After the test tube is inserted into the water, only you are removing your thumb. So see, no single air bubble here. It is completely filled with water. Okay, no single air bubble. There is no gas at the end of the test tube. Now, you take this setup and keep it in sunlight. In sunlight and observe. For some time you need to observe it. See, the gas bubbles are coming. You can see this hydrilla plant has accumulated some gas bubbles. And these gas bubbles, since they are lighter than water, they move to the top region. Okay? They move to the region top. See, one by one the bubble is escaping out to the top region and slowly the gas will be collected at the end of the test tube. So this is the gas collected. When we started the experiment, there was no gas here. Now you can see there is gas. So after the gas is collected in a sufficient amount, when you are removing this test tube also, you need to be very careful, because if any other gas enters into the test tube, your experiment is a failure again. So you are taking out this test tube, inverting it carefully, and we need to introduce a burning splinter into this to test that this gas. See, he has taken a burning incense stick. You can take anything that burns and open your thumb. It burns vigorously. Let us see again what happens. He brought it near the test tube and see how does it burn. It burns vigorously. So we should not remove this thumb unless our burning splinter is ready, because that is a very small amount of gas. If it escapes, our entire experiment is wasted. So this is the experiment, children. We have 2 more experiments. So the next experiment we have is experiment to prove that carbon dioxide is essential for photosynthesis. Okay, we have proved that starch is produced during photosynthesis, oxygen is liberated during photosynthesis. Now we have an experiment to prove that carbon dioxide is required for photosynthesis. Okay, the simple trick we are going to apply here is we'll supply one part of leaf of same plant with carbon dioxide, the other part of same leaf we will not allow it to take carbon dioxide. How is it possible? We'll see now. So we have a solution which is called as potassium hydroxide solution. We can take it in the form of pellets also. When you take it inside any container like this, all the carbon dioxide that is present in this container will be absorbed by potassium hydroxide or KOH. Okay. So in this way, if you put one part of leaf inside this bottle like this, if you insert it partially into a bottle, this part of leaf will not get carbon dioxide. The other half of leaf which is outside will get the carbon dioxide. This part will not get carbon dioxide. This part will get carbon dioxide. Okay, that is the trick. So after that you can test for starch. If the starch is produced on this side, it proves that carbon dioxide is essential for photosynthesis. If it is produced this side, we can prove that carbon dioxide is not essential for photosynthesis. Okay, but the problem is leaf already have starch. Even if you have provided this environment where half leaf gets carbon dioxide, half leaf does not get carbon dioxide, leaf will already have carbon dioxide. This part which is not getting carbon dioxide already have starch in it. So we need to do something to remove that starch. What can we do? We have a simple trick for that. We'll take this potted plant and keep it in a dark place for 1 week. Okay. What will happen if you do like that? See, in dark place, plant will not get any sunlight to make its own food. So what it has to do is it has to utilize all the starch that is present in its leaves for its daily metabolic activities, whatever is going on. So for that, after 1 week, if you remove that, leaves will be now free from starch. Okay, then you can go with this experiment. We'll start from the beginning. What are the materials required for this experiment? So the materials required for this experiment are a potted plant which is having leaves. This leaves should be so thin and long that it can be easily inserted into this glass bottle. Okay, or you can take the glass bottle itself wide enough and rubber cork also wide to fit that bottle. That is a little problematic. So we can take a plant which is having thin leaves. Apart from this potted plant, we need potassium hydroxide in the form of pellets or in the form of liquids. This is a glass bottle. This is iodine solution, grease. We also need these all things for performing the experiment. Yeah. We need a split cork also. A split cork is a cork which is having a split in the middle. So that leaf can be inserted from that into the bottle. Okay, that is split cork. So we'll take this potted plant which is having thinner, long, narrow leaves, and we'll keep it in darkness for 1 week days, so that all the starch that is present in this plant will be gradually removed in this week time. Okay, after that, when you take out after 1 week, this plant will be completely free from starch. So now we can perform the experiment. What we need to do is, we need to take a glass bottle with 5 to 6 ml of KOH solution in that. Okay. Then a split cork is fixed to the leaf, and this split cork is inserted along with leaf into the bottle. See, KOH solution is here. In this bottle itself, we are fixing this, and this bottle should not fall off. We are keeping it on it. Support. After that, there should not be any. Why we put this rubber cork? So that there is no entry of carbon dioxide from outside to inside. First thing. But still, through the split, any carbon dioxide should not enter into that bottle. For that reason, we are greasing it. So when you apply some grease is nothing but an oily substance. So it does not allow any entry of air molecules, atmosphere molecules into that bottle. Okay. So now we have a perfect environment where part of the leaf gets carbon dioxide, part of the leaf does not get carbon dioxide. Okay. We leave this setup in sunlight. This setup should be left in sunlight for few hours, 4 to 5 hours. We leave it in sunlight. Afterwards, we will detach this leaf from this potted plant and separate the leaf. After the leaf is separated, we should go for starch test. So the entire process, taking the methylated spirit in test tube, boiling it in water bath, removing all the chlorophyll from the leaf, will be the same process as we saw in the starch experiment. After that, leaf becomes pale white in color, and once the leaf is in pale white in color, then you can go for iodine test. Put 1 drop of iodine. You need to spread it on the entire leaf now, not in the center itself, because we need to understand which part of leaf has performed photosynthesis and which part of the leaf could not perform photosynthesis. So we are applying this iodine solution uniformly on entire leaf and leave it for few minutes. 1 or 2 minutes is enough for this iodine solution to react with starch and turn it into bluish black color. So the result is like this. The part of the leaf, this is the part of leaf which is inside the bottle, there is no color change. But the part of leaf which is outside the bottle, you can see clearly there is a color change, that is, it turned into bluish black. So this experiment proves that carbon dioxide is required for photosynthesis. And at the last, we have another experiment to prove that sunlight is required for photosynthesis. So when you do this experiment also, you need to do the same thing. You need to keep the plant in darkness for weekdays so that the starch present in the leaves is removed off. So what are the materials required for this experiment are a potted plant, a clip, a piece of cardboard paper and iodine solution. So take the potted plant. See, now you should take broader leaf because we are going to arrange the cardboard to this leaf. So you need to keep it in dark place for 1 week, weekdays, so that you can de-starch it. After 1 week, remove the plant from darkness and now the leaves are free from starch. And this is a very simple experiment. You need to just put the cardboard, fix it with the help of this clip so that this part does not receive sunlight. Okay, we have a condition now where part of leaf is receiving sunlight, these two sides, and part of leaf is not receiving sunlight, that is in the center. We'll keep this setup in the sunlight. See, all other things are common. We'll water the plant. Equal water is obtained from different regions of this plant. The amount of chlorophyll is same, carbon dioxide taken is same, even if it is a cardboard. See, if you have space here, there is a chance for carbon dioxide to peep even from the lower side where stomata is present. So only difference is a part of leaf gets sunlight, a part of leaf does not get sunlight. Okay, carefully we will remove the cardboard. We will perform the same starch experiment where methylated spirit is taken in the test tube and water is provided in the beaker. It is boiled and chlorophyll is removed from that, and then iodine test is performed over this leaf. So as we have applied uniformly in case of carbon dioxide experiment, in this experiment also iodine need to be applied uniformly throughout the entire leaf. Okay, the entire leaf should be applied with iodine and left for few minutes. Let's see what's happening. So this part turned into bluish black color, this part turned into bluish black color, but not this part. Okay, there is a small like this part has turned into bluish black color because there are veins. Veins will pass the food material, right? Xylem and phloem will pass the food material, whatever is produced here and there is a passage, that's all. Okay, but here it is clearly showing that this part did not participate in photosynthesis only. So this is an experiment which proves that carbon dioxide is essential for photosynthesis. Sorry, sunlight is essential for photosynthesis. Okay. Okay children, that is it for today. I hope you have followed it. Keep yourself safe, healthy and also study well children. Bye.